Hello, today's lecture we're going to focus on concentration gradients and tonicity. In order to understand this material, it is important that you really understand diffusion. So if you haven't got a good understanding of diffusion, I do recommend that you go back and watch the diffusion video. You go back and read your notes on them. But it is important that you understand diffusion in order to understand concentration gradients and tonicity. Because now we're really going to talk about why diffusion is important, especially to us, our human body. So in order to understand that, we first need to understand that all our cells have a cell or plasma membrane. So you can either hear the name as a cell membrane or a plasma membrane, but in either case they're talking about the same structure. And this plasma membrane is really going to control what goes in and out of your cells. And it's what we call a selectively permeable membrane. So in order for something to get through something, through, let's say, a wall or a membrane, like our cells, they have to be permeable. So when something is permeable, we mean that something is able to diffuse across a membrane. If something is unable to get through, then we call that impermeable. However, the cell membrane or the plasma membrane is what we call selectively permeable. And we call it selectively permeable because it's really going to help control what goes in and what comes out of our cells. And it's going to do this, generally speaking, with most of the things that we come across with. There are a few things that your cell membrane is not as good at controlling, and one of those things is water. And I do want you to keep that in the back of your mind because that's going to play a big role in a couple slides from now. Now, before we move on, there is some basic vocabulary that we do have to understand in order for us to proceed. And so we're going to talk about what a solute is. And a solute is just a substance that you can dissolve into something. It's whatever you're going to mix in. And the solvent is the substance that dissolves the materials. So in most cases, especially in biology, that solvent is going to be water. So it's whatever is going to do the dissolving. And of course, we have a solution when we have a mixture of a solute and a solvent. So for example, if we have Kool-Aid, we know that it's a mixture of a couple things. It's going to be water and Kool-Aid powder. We do obviously need our sugar because, you know, who likes their Kool-Aid without sugar? And it has to be a lot of it. But usually when we talk about that example, then our sugar, our Kool-Aid powder, those are the things that we're dissolving into the water. So because those are the things that we're mixing in, then those things, the sugar and the Kool-Aid, would be our solute, whereas the water would be our solvent. Of course, that refreshing beverage that we then drink is the solution. Now, we're going to talk about concentration gradients. And the first one we're going to talk about is something that is isotonic. Now, when we talk about these gradients, obviously we have to have something to compare it to. So when we're talking about gradients and when we're talking about these, whether it's isotonic, hypertonic, or hypotonic, then it's going to be easier for us to understand that we have to be able to compare these items to something else. So if we're talking about something being isotonic, it's always isotonic to something else. It's not okay to just say, well, this is isotonic. Okay, well, this substance A is isotonic, but it's isotonic to what specifically? So remember that we're always going to be comparing a couple of things. Isotonic just means that the solution is going to have the same concentration of solute, and in isotonic solutions, we end up with no net movement of water. So here's a depiction of isotonic solution. Our solutes are in green, and it is, in this case, we're just going to have to understand that most of this environment here and here is water. So water is actually going to move in to the cell, but it's also going to move out of the cell freely because, like I said earlier, the plasma membrane is really good at controlling what goes in and out of the cell, but not necessarily with water. Water can, for the most part, on most occasions, it can move freely across the plasma membrane, and that's what you see here. So water is going to be moving across at the same rate because we have an isotonic solution. The inside of our cell, which is here, is the same as the outside when we're talking about the concentration of our solutes. So we have these in green. Those are our solutes. And we have the same concentration on both inside and outside of the cell. Our next movement, or our next category that we're going to talk about, is hypertonic. So let me go ahead and just get rid of this. Sorry, the import didn't quite work like I wanted it to. Now, if we're going to talk about hypertonic, this prefix hyper means above. It means a lot. And the reason we call it hypertonic is because hypertonic solutions have a high concentration of solute. And when we have a hypertonic solution, water is going to move into that solution. And we're going to talk about why in just a few minutes, but I do need to show you this diagram here. Oh, too far. So here's a hypertonic solution. So again, here we have our cell. And here's the outside. Now, inside of our cell, we have cytoplasm, and it contains a lot of solutes. There's lots of proteins, lots of stuff that's dissolved into the cytoplasm. And so we do have some concentration of things in there. Now, when you place a cell in a hypertonic solution, it's important that you understand that water is going to move out of the cell into the hypertonic solution. Water will move into a hypertonic solution. Now, a hypotonic solution is the opposite. So again, let's look at this prefix here. We have hypo, which means low. Hypo is always below normal. So solution that contains a low concentration of solute is going to be called hypotonic. And when we have a hypotonic solution, water is going to move out of that hypotonic solution. So let's look at an example again. Let's go back and look at our cell. So now here again, we have the inside of our cell, and we can see our solutes all inside. And outside, we don't have that many solutes. We have one. So this is outside of the cell. And when we place a cell in a hypotonic solution, what we're saying is that that solution we've placed the cell in has a lower concentration of solutes. Therefore, it's hypotonic. So that solution out here is hypotonic. And when that's the case, water is going to move away from the hypotonic solution. In this case, it's going to go inside of our cells. So this is opposite of what happens with hypertonic, where the water is going to leave. In hypotonic solutions, water is going to enter because it's going to move away from the hypotonic solution. Now, we can actually see this, and the reason we're going to talk about this is, again, because the plasma membrane will prevent most solutes from diffusing in and out of the cell. So the concentration of solutes doesn't really affect the permeability of the plasma membrane, but it will affect the movement of water. So here we can actually demonstrate, this is actually an experiment that was performed in a laboratory. And so if we have on one side a highly concentrated solution, and then on the left side, a lower concentrated solution. Okay. Now we have something to compare it to. We now know that this side, we're going to call it side B, is hypertonic to side A. We're going to call this A. Because side A is hypotonic to side B. Now, when this happens, remember, water is going to move from hypo to hyper. So if we know that side A is hypotonic, then we know that water should be moving this way. And when we perform this experiment, because what we're saying is that in the middle here, they've placed this membrane that prevents the solutes from diffusing away or across that membrane. So you can't have that solute moving across it, only water. So this is a selectively permeable membrane. So only allowing water to move through. So because we have a hypotonic solution and a hypertonic solution, the solutes are going to remain on the same side, but water is going to move from the hypotonic solution to the hypertonic solution. So here's our water levels to start off with, and they're pretty even, but now our levels are no longer the same. Okay. So after some time, that water is going to move from that hypo to that hyper. So again, here's looking at that semi-permeable membrane. You can see all the water molecules. Okay, they're going to move from an area of high concentration to low concentration. So why does this matter? Okay, there's going to be a couple problems that we can run into when we talk about tonicity. We want equilibrium. Those cells are going to be greatly affected if we change the concentration gradients in the environment. So it's going to matter. We have to determine our tonicity in comparison in relation to our cells, and then we're going to have to talk about and determine where that higher water concentration is. That's what what the brackets mean right here. It's just concentration. Of water. Now, when we have a hypotonic solution, what that means, if we recall our vocabulary, it means that it had a low solute concentration. So the first thing that we had decided was, first we have to determine our tonicity. So we know that inside of the cell it's a higher concentration, and outside of the cell it's a lower solute concentration, because again, if it's a hypotonic solution, it means that there is a lower concentration of solute outside of the cell. So the first thing, we establish those gradients. So in our solution, we have two things. We have a solution and we have a cell. Now, we are given this hypotonic solution right there. So that tells us that we have a low solute concentration in my solution outside of the cell. Okay. It also tells me then that inside my cell, because the solution outside of the cell is hypotonic, that makes the inside of my cell hypertonic. So that makes this a high solute concentration. Now the question then becomes, can these solutes move freely across the membrane? And the answer to that question is no. The solutes cannot move across the membrane freely. Only water can. So what we're interested in is water. Now we're doing this, establishing our gradient first, because that will tell us where our water concentrations are. So if we have a low solute concentration, then it's always going to be opposite. That tells me that there is a high water concentration in my solution outside of the cell. Well, if that's outside of the cell, then inside the cell that I have a high solute concentration, then by default I should have a low water concentration. So now that I've established my water concentration and I know water is going to move, then I know water is going to move through osmosis, which means it's going to move from an area of high concentration to low. I know my high concentration is here, so it's going to move from the solution into my cell. So all that water is going to be moving out, and just like cells, just like a balloon, the cells are going to fill up and fill up and fill up and fill up and fill up with water until they explode. Now, when I have a hypertonic solution, I still want to follow the same steps. So if I want to know what's going to happen to the cell, I have to first establish my gradient. So I have inside of the cell here, intracellular, so we have a cellular environment, and then we have an extracellular environment, or outside of the cell. So we're going to go ahead and establish those two things. So I'm going to make my little chart. I'm going to have outside and then I'm going to have inside. Now I know my outside of the cell is hypertonic because that was given to me. I have a high solute concentration on the outside, so that makes my outside hypertonic. Now because the outside is hypertonic, then that by default means that my inside of my cell is going to have less solute dissolved in the cytoplasm, so that's going to be hypotonic. So I've established my gradients. So then I have to ask myself, what are my solute concentrations? So in a hypertonic environment, that means I have a high solute concentration. And in a hypotonic environment, I have a low solute concentration. Now, because I know my outside is hypertonic and that it has a high solute concentration, then by default, if there's a high solute concentration, then there's going to be a low water concentration. And then on the inside, if I know I have a low solute concentration, then by default, I'm going to have a high water concentration. Remember that all of this is relative, okay, in relation to each other. So the outside, high solute concentration, low water concentration. Inside, low solute, high water. So what does that mean? We know that the plasma membrane is going to prevent solutes from passing freely through the membrane. So we know the solutes are not going to diffuse from high to low. However, water can move freely from inside, outside to inside. We just got to know what direction it's going to move. Now I know my inside of the cell has a high water concentration and the outside has a low water concentration. So I know my water is going to move this direction, okay, from inside my cell to outside my cell. And because I'm having a decrease of water from my cell, all that water is leaving, and the more water that leaves, the smaller that cell is going to leave. It's gonna be, and so it ends up shrinking your cells and killing them. And as the cells shrink, we call that crenation. And so the cells crenate and they die, they shrivel up. So that happens again because it's a high solute concentration outside the cell, low water concentration. So again, here are pictures. If we place a cell in a hypertonic solution, the cell is going to crenate, it's going to shrivel up, it's going to die because water is going to leave the cell from a hypotonic environment inside the cell to the hypertonic environment outside of the cell. In a hypotonic environment, then again, the cell's still going to die, but instead of dying because it's shriveling up, it's going to swell up, it's going to swell, and it's going to lyse, it's going to burst. Because it's going to fill it with too much water that it won't be able to hold its shape and it's just going to burst out, much like overfilling a water balloon. And so, again, because the water is moving from the hypotonic environment outside of the cell into the hypertonic environment inside of the cell. And when we have an isotonic environment, which is what our blood is, it's our blood, the liquid that makes up our blood, the plasma, is actually isotonic to our red blood cells and to our white blood cells. Again, we want it to be that way because when it's isotonic, we have the same water movement inside and out of the cell so that the cells aren't affected, at least the shape isn't.